Do you know what lactase is? No? That's what I thought. Today we are going to discover exactly what lactase is and what in the world it does. Lactase is an enzyme normally produced by the human body to catalyze the breakdown of lactose into glucose and galactose. However, there are some cases where the body does not produce this enzyme, therefore leading to lactose intolerance because the body is unable to digest this disaccharide. Let's take a closer look at lactose. That's too close. That's better. What is lactose? Well son, I'm glad you asked. Lactose is a disaccharide sugar found in dairy products such as milk. The two components of this molecule are glucose and galactose, both of which are monosaccharides. These two monosaccharides are held together thanks to dehydration synthesis. Monomer plus monomer equals polymer plus water. So how on earth does lactase break this bond? Easy peasy. Lactase is an enzyme. Enzymes have an active site. The active site has the shape of a specific substrate, which helps the substrate bind to the enzyme. In this case the substrate is lactose. Think of it as a lock and key. Once a lactose molecule binds to the active site on the lactase, hydrolysis occurs and the lactose is broken down into its simpler sugars. In case you've forgotten, they're glucose and galactose. Whoa. Let's back up a bit. If humans cannot produce their own lactase, they will have trouble digesting lactose. Luckily, a group of MLG scientists invented the lactase pill, which is now commonly available in drugstores around the world. Since there is nothing else to do, let's do a little experiment instead. How does heat affect the rate at which lactase breaks down lactose? I don't know. Let's find out. We will be studying the effects of heat on the reaction rate of lactase and lactose. I predict that the hotter it gets, the faster it will get as well. Likewise, the colder it gets, the slower the rate, mate. For this experiment you will need the following. 3 15 by 125 mm test tubes. 2 500 ml beakers. Ice. 1 hot plate. Lactase tablets. 100 ml of milk. Glucose test tape. Stopwatch or timer. A thermometer. A glass stirring rod. A dank test tube rack. A test tube holder. Tap water. A mortar and pestle. MLG procedure. Fill each 500 ml beaker half full with tap water. Add ice to one of the beakers and place the other beaker onto the hot plate. Turn on the hot plate and add a thermometer to each beaker. Fill each of the test tubes halfway with milk. Place one of the test tubes inside the ice beaker and another inside the heated beaker. Leave the last one on the test tube rack. Use the mortar and pestle and crush the lactase tablet into powder. Record the temperature of each of the test tubes. Now, spread the lactase powder evenly across the test tubes and stir each solution with a stir rod. Begin the timer and place a strip of glucose test tape in each test tube. Record whether or not the test tape changes color. Every 5 minutes, place another strip of glucose test tape into each beaker and record your results. Continue until all of your test tapes change color or you run out of time. Let's take a look at an example of this lactase in effect, shall we? Here, we can see our beautiful stomach and part of our small intestines recreated in the best game ever, Counter-Strike, Global Offensive. In this first scene, we see the innocent lactose sugar after being ingested, traveling down past the stomach and into the duodenum, where it meets with the lactase produced in the intestinal lining. When the lactose comes in contact with the lactase enzyme, the lactose latches onto the active site of the lactase. The lactase enzyme then, using hydrolysis, does its work in breaking apart the lactose into its basic components, glucose and galactose. The newly broken down molecules now travel down the intestines, able to be digested. In this second example, however, the lactase is not lucky enough to have an ideal temperature in the intestines. The lactase enzyme is therefore denatured because of the high temperatures, causing the molecule to change shape. Unable to attach to the active site of the newly altered enzyme, the lactose passes through the intestines unmodified and thus unable to be digested. Given the fact that we are not perfect, Our results were inconclusive to say the least. Unfortunately, none of our glucose test tapes changed color over the 40 minutes of testing. This may be due to many reasons, such as leaving the test tape in the solution for too long, or maybe the temperature of the solution made it difficult for the tape to change color. It could simply be a problem with the materials we used or the temperatures we decided to test. Perhaps the glucose test tape was expired, or the milk did not have any lactose in it. Maybe all the temperatures we tested were out of the range where the enzyme functions. Even though our results were uncertain, I can tell you the results that should have been obtained based on the research we did. The lactase enzyme functions best near or slightly above room temperature, at roughly 20 to 50 degrees Celsius, while being optimized at 46 degrees Celsius. This is because colder temperatures slow down the function of the enzyme, while very hot temperatures will denature it. The temperature of the human body is 37 degrees Celsius, making the lactase enzyme very effective in humans. This means that the fastest reacting test tube should have been the one at room temperature, at 21 degrees Celsius. To improve this lab, we would choose to test a larger variety of temperatures. If possible, we would also use more samples in case something goes wrong with one of them. Although our MLG experiment did not produce the results we were hoping for, we were able to explore the inner workings of the lactase enzyme.